Hello, this is Mr. Wartsky, and we are going to start our very first screencast on meiosis. Remember, if you have any questions, you can watch the video over again, listen to or ask me questions, or read the textbook. So let's get started with meiosis. So in higher organisms, cells are diploid. D2N. Now that is the abbreviation for diploid 2N, which means double set. Why is it a double set? Because one set comes from mom and one set comes from dad. Gametes are haploid. That means single set. And this is represented by the letter N, which is half of a diploid. So mom gives you N chromosomes, dad gives you N chromosomes, and you end up with N plus N, which is 2N. In Hugh people, we have a diploid number of 2N equals 46, and that means that our haploid equals 23 chromosomes. Remember that there are two types of cells. There are somatic cells, which are body cells, and gametes, which are reproductive or sex cells, specifically sperm and egg. So we have 46 chromosomes per cell. Each chromosome is a single strand of DNA, and on that DNA strand we have hundreds to thousands of genes, which are locations that undergo protein synthesis to produce proteins. A locus is a specific gene location on a chromosome, and we can get pretty good at defining what genes are produced where on which chromosomes. In Hugh people, since we have 46 chromosomes, we really have 23 pairs of chromosomes. 23 pairs. Each pair, they are called homologous chromosomes. Now, homologous chromosomes are a tough concept to understand, so let's go through this and make sure you understand that they are chromosomes that carry the genes for the same traits. They don't have to be the same genes, but they are genes for the same trait. Now you get one of those chromosomes from mom and another chromosome from dad. So you get two chromosome number ones, one from mom, one from dad. And on that chromosome number one, you will have the same traits. And those will be the genes that code for the proteins that give you those traits. But they don't have to be the same gene. So we have one chromosome from mom, one chromosome from dad. This is before DNA replication. So you have two chromosomes here. DNA replication occurs, or DNA synthesis, and you know what happens in DNA synthesis. And we end up with now two chromosomes, but each chromatid is made up of, chromosome is made up of two chromatids. So we have a chromosome here, and each one of these things is an exact copy of the other. This is an exact copy of this, so that we say that this is one chromosome, this is one chromosome. So we still have two chromosomes, but each chromosome is made up of two chromatids. Hopefully that makes sense. So homologous chromosomes, we have one chromosome from mom, one chromosome from dad. I think I've said that like 4 times now. They contain genes with the same traits in the same location. That's the second time I said that. There are 22 homologous pairs. Those are called some body, they code for the body stuff, all the proteins. And then we have one pair that is not homologous. Those are the sex chromosomes. That's the X and the Y chromosome. In meiosis, the homologous chromosomes will pair up, exchange information, and then separate. Pair up, exchange information, and then separate. So here this is homologous chromosomes pairing up. They get the center mirrors the region, the kinetic the actual structures. These are chromatids here, and they're paired up. Now they pair up, they actually touch each other. If they touch each other in the right spot, they pair up. So when they pair up, we're going to talk about exchanging information, and that is called crossing over. Remember we talked about crossing over? We talked about mutations, and all those things happen because of either unequal crossing over or things floating away or things getting flipped around. What happens is that the chromosomes are actually cut, and they are resealed to the other homologous chromosome. So the DNA is actually exchanged. Lots of stuff can happen during that exchanging, but most of the time things go really well. So we have two chromatids here that are not recombined, and 2 chromatids that are recombined. And notice that, therefore, each and every chromatid is different than the other. So in mitosis, you're going to get this chart down. So let's write this down. We have the characteristic meiosis ploidy means the number of chromosomes. So mitosis can happen in haploid, diploid, and polyploid cells. Meiosis, it can only happen in diploid and polyploid cells. The number of nuclear divisions in mitosis is 1, meiosis is 2. And the chromosomes after the process in mitosis, the chromosomes are identical to the parent. Whereas in meiosis, they're unique. Why? Because of the crossing over. And I want you to draw the crossing over, by the way, so go back and draw it. So there are 2 stages of meiosis, and they're cleverly called meiosis 1, which is reduction in the number of chromosomes. We're dividing the chromosomes by 2. And meiosis 2, which is division of the chromosomes, in which we split the sister chromatids. So we're going to get into the steps of meiosis 1 and meiosis 2, and I know you can't wait because I cannot. Here's the overview. Now the phases are called prophase, metaphase, anaphase, and telophase. Sound familiar? I hope so. Except we're missing prometaphase, but that's okay. We're just taking prometaphase and prophase and just slamming them together there. Okay, so here is an overview of meiosis. You don't have to get the picture down because you'll be drawing the pictures as we go along. So this is meiosis 1, that's what M1 stands for. And this is interphase 1, that should be an I1, but they look like II or LL, but it's I1. We have interphase 1, which is G1S and G2. And you know what G1S and G2 is? Right. Okay. And this is the cell that I want you to draw. Draw that. You have a circle and a smaller circle. I think you can handle that. Now, in meiosis 1, prophase 1, this is the longest and most complicated of all the steps of meiosis. So if you understand this step, then everything else is a slice of pie. So prophase 1 starts off similarly to prophase in mitosis. The chromosomes condense because of MPF. The nuclear membrane fragments because of MPF. The nucleolus disappears because the chromosomes condense, and the spindle fibers form. And all of that stuff is from prophase 1 of mitosis. But here's the new stuff. The homologous chromosomes pair up by touching each other, and if they touch each other in the right spot, then they bind together. They form what is known as a tetrad. Tetrad? Why a tetrad? Well, tetra means 4, and if we have 2 homologous chromosomes together, you will have 4 chromatids. That's why it's called a tetrad. Now, the 2 homologous chromosomes are held together by the synaptonomal complex. In prophase 1, there is an exchange of DNA, which is known as crossing over. The chiasmata is the point where the crossing over takes place, and this is known as sex because the chromosomes or the DNA is being mixed up. Now, I hope you understand the difference between chromosomes and chromatids. Remember, chromatids are copies of chromosomes. Here is a picture of prophase 1. Now, you only have to do 2 chromosomes here. So we have 4 chromosomes, 2 here and 2 here. Notice they're crossing over here, crossing over there. Beautiful. Here's the nucleus. Hopefully it'll be breaking down soon. But draw prophase 1. That was really quick, wasn't it? Draw prophase 1. Hit pause. Then we have meiosis 1, metaphase 1. The kinetochore spindle fibers lengthen and shorten so that the synaptonomal complexes line up on the metaphase plate. This is a checkpoint, remember, because if they're not lined up, it will not continue down meiosis 1. Now, APC, or anaphase promoting complex, is also produced. So right now, if you take a look at here, we will have, right, we have 4 chromosomes here. 1, 2, 3, 4. Right? 4 chromosomes. The synaptomal complex should be in the middle here. Okay. And what's going to happen is in anaphase, they're going to divide. and we're going to end up with 2 chromosomes per cell. So we have 4 chromosomes. How many chromatids do we have? 8. 4 chromosomes, 8 chromatids. Draw that picture. Hit pause if you need to. Notice the kinetochore spindle fibers right there. Don't forget the kinetochore. You draw the polar going back and forth, and here's the aster spindle fibers there. Good picture. Good picture. Anaphase 1. The APC, or the anaphase promoting complex, will digest the synaptonormal complex. The kinetochore spindle fibers short and homologous chromosomes separate. So what happens to the number of chromosomes when anaphase 1 happens? They are divided by 2, or they are reduced. So here, let's take a look here. So again, ignore 2 of these things right here. So we'll have 1, 2, 1, 2. So 2 chromosomes go this way, 2 chromosomes to go this way, but you had 4 chromosomes here to start with. Now each chromosome is made up of 2 chromatids, but we still have 2 chromosomes going to one side and 2 chromosomes going to the other side. Draw this picture without these 2 chromosomes there. Make sure you have it crossing over, right? Hit pause. In telophase 1, the chromosomes decondense, the nuclear membrane reforms, the nucleus reappears, the spindle fibers become disorganized, and we have cytokinesis. So again, the chromosomes still have 2 chromatids, but the chromosome number has been halved in each of the cells. And here's a picture. We have 2 chromosomes here, 2 chromosomes there. We started with 4 chromosomes made of 8 chromatids. Now in each one of these cells, we're going to have 2 chromosomes made up of 4 chromatids in telophase 1. Hit pause, draw the picture, please. Draw the picture, hit pause. Cytokinesis 1, cell divides. We have reduced the number of chromosomes, but each chromosome has 2 chromatids. And we are going to stop after meiosis 1 today. We'll continue with meiosis 2 in the next screencast. Have a good night.